In this video, we're going to talk about viruses and bacteria and the genetics of viruses and bacteria. And this is important because viruses and bacteria are both important in biotechnology and recombinant DNA, and also in a lot of the discoveries that have been made. We talked about Hershey and Chase and Griffith and transformation. So we're going to look at now sort of the structure and genetics of viruses and bacteria. And here you have a virus. You can see it's very small, just in the sort of even below a micron level. It's actually in the nanometer level, really small. And bacteria, again, being about 1 micron or so as compared to maybe the average eukaryotic cell, which might be like 10 micrometers or something like that, much larger. So we're talking about very small structures, very simple structures, but because they are so simple, we can learn a lot about them and their genetics. So let's look at viruses first. So we have different types of viruses here. They all have different shapes, but they all have sort of a common characteristic, and that is some kind of protein coat or capsid or capsule, right? And then either DNA or RNA inside. So here's a bacteriophage. This is what Hershey and Chase used to find out that DNA was being injected by viruses into bacteria, right? Here's a influenza virus, which causes obviously the flu, and a couple other commonly studied viruses there. Again, all have either DNA or RNA inside a protein capsule, right? And we know it's the DNA that goes into the host cell and takes over cell activities. Well, there's two ways that can occur. The first one is called the lytic cycle. So lytic means that the term comes from lysis, which means to break up or dissolve. So what happens is we've got our virus, and the virus recognizes a specific cell, inserts its, in this case, viral DNA, sometimes RNA. Okay, two things are going to go on there. First off, transcription, and the viral DNA is going to be transcribed into viral proteins, those, again, capsid proteins, or those proteins that are going to be on the outside of the virus. And we're also going to replicate the virus, the viral DNA, so that we can then assemble the DNA into these proteins, make new viruses. The cell fills up with these viruses and eventually lyses, hence the term lytic. So this cycle is immediate. As soon as the DNA is injected, it takes over cell activities, cell fills up with viral components, they get assembled and rupture and leak out into the cell. Okay. So that's what the way a lot of cells or a lot of viruses function, like rhinovirus, which causes the cold, influenza, which causes obviously the flu. But there's another strategy some viruses use. So over here on the left, here's the lytic cycle, and we can see taking over the cell, immediately producing and lysing the cell and producing and releasing more viruses. But sometimes that strategy is not the best. If you want to really kind of be successful, sometimes you don't want to immediately kind of have this massive assault. You want to kind of lay low. So there's another strategy many viruses use, and that's what's called the lysogenic cycle. So lyse, again, means to split or break up, but in this case we're breaking up the genes, the genome of the host cell. So in this case, what happens is the virus recognizes a host cell, inserts its DNA or RNA, okay, but that then gets lysed or inserted into the host's DNA. In this case, it's a bacterial cell. Notice the circular bacterial cell, bacterial DNA. And that bacteria will continue kind of unaffected. It will grow, it will divide. Each time it divides, it's going to copy that viral DNA and pass it on to future generations. So what we end up happening is a lot of a lot of bacteria now have this viral DNA in them. And at some point, some trigger is going to cause this viral gene to become lytic. It's going to flip into the lytic cycle, and it's going to start to produce viral proteins and viral DNA and assemble them into viruses and lyse the cells or release the viruses. So an example of this in humans is the herpes virus, where someone will get herpes, they'll carry that virus, it will remain inactive or dormant for long periods of time, and then all of a sudden they'll have an outbreak and they'll have sores and there'll be massive releases of viruses at that point when their virus is in that lytic stage. So if we look at HIV, HIV is called a retrovirus. So retro means going back. If you're into retro music or something, you're going back or looking back. And retroviruses work in reverse. Instead of going from DNA to RNA to proteins, These are RNA viruses. Notice they contain RNA, and they inject that RNA into host cells, but they undergo a lysogenic process, right? So they have to then convert that RNA into DNA. And the way they do that is with an enzyme called reverse transcriptase. So what does this do? Well, this does transcription in reverse. It takes an RNA code and it matches complementary DNA bases to it, so that when we are done, we've converted that RNA, viral RNA, into DNA. That DNA can then go into the nucleus and be inserted into the host cell. So again, HIV is a retrovirus. It also undergoes this lysogenic process where it inserts its DNA. That's why HIV takes many, many years before it becomes active. When people get exposed to HIV, they have many, many years of sort of normal life where they can live and even spread that virus, right? Because it's in this sort of inactive stage. At some point, that provirus will initiate the lytic cycle where it will start to produce RNA. That RNA will then transcribe the proteins and copy itself into more HIV viruses, which will then be released and spread to other cells. In this case, they're attacking a specific cell in humans called T cells. T cells are an important component of our immune system. So HIV works by really destroying our immune system, the T cells in our immune system, which are involved in fighting off all infections. So that's how a retrovirus works. It's RNA that gets converted into DNA, that gets inserted into the host genome, remains dormant for long periods of time before it becomes initiated into a lytic response and destroys the cell. Prions are sort of an oddity, and we'll just talk about them here since we're talking about viruses and bacteria. Prions are not living. They are proteins, and they are infectious proteins. And good example of them is mad cow disease. You've obviously heard of that. There's a human equivalent to mad cow disease called Creutzfeldt-Jakob's disease, which basically does the same thing to a human brain that mad cow does to the cow's brain, and there's a concern that the mad cow disease could be transferred from cows to humans if we ate contaminated meat. And this is scary because what these prions do is they basically get into an environment and they just convert other proteins into prions. They are infectious, so they're capable of converting proteins into prions, which then can convert other proteins into prions. And the problem with this, especially with cows affected with this, is these prions also tend to be resistant to heat. They don't get denatured the way most proteins do. So you can't just cook them and kill them. So it's a big concern when they discover animals affected with prions, because these proteins are very stable, hard to destroy, and highly infectious. But fortunately, over the past 10 or 15 years, we haven't heard anything about any new outbreaks of any prion-related diseases. So now let's talk about bacteria. So bacteria are asexual, right? They have one big circular piece of DNA. They sometimes have other little small circular pieces of DNA called plasmids, which we'll talk about. We talk about recombinant DNA in biotechnology. But they're asexual, they're simple. They can basically replicate that DNA, so they can replicate their chromosome, and then they can move the chromosome to opposite regions of the cell, pinch in half, and produce identical cells. This process is called binary, meaning two. Fission is splitting one and making two, like nuclear fission splits an atom into two smaller atoms. Binary fission is basically a bacteria replicating its chromosomes and splitting into two. And this is an easy process. It's fast. Bacteria with plenty of food in an optimal environment, temperature, moisture, food, they can do this about every 20 minutes. So this is a very efficient and fast way to reproduce. Unfortunately, it doesn't allow for any genetic recombination, right? So you're not going to get a lot of variation. You're not going to allow the organisms to evolve very quickly in this asexual mode. So how do bacteria allow for more genetic variation? They've been around for 3.5 billion years, and they have evolved. They have changed. We're going to look at some mechanisms that they use to evolve and change. The first one is called transduction. Trans means across, transduction, moving from one to another. So what can happen is viruses, when they insert their viral DNA and they cause the production of viral components, oftentimes make mistakes and are sloppy. And sometimes, as the new viruses get assembled from a bacteria, they have some of the bacterial DNA in the viruses. And then when that virus attacks a new bacteria, it's going to inject that bacteria from the previous bacteria into the new bacteria. So what viruses sometimes do accidentally is introduce DNA from one bacteria into another. When they do this, it's called transduction. And many harmless bacteria have become very pathogenic and very dangerous because they've had genes moved from other bacteria into them by viruses. And this process is called, again, transduction. Transformation, that should sound familiar because when we talked about Griffith and how he discovered this transforming factor, he discovered accidentally this process of transformation where the pathogenic dead bacteria were able to somehow give something to the harmless living bacteria to make them very deadly. Well, what they were able to give them was DNA. So we have in bacterial cells, occasionally bacteria can leak out of the cells, and bacteria have evolved the ability to, in their cell walls and cell membranes, to take in foreign DNA. And we're going to look at a lab where we do this. We're going to try to get bacteria to take in a glow gene that's going to make the bacteria glow through this process of transformation. And they can take it in and they can incorporate it into their genome. And again, what does this do? This allows bacteria to sort of take on new variations and more genetic variation by assimilating bacteria from their environment, from other bacterial DNA that are just sort of floating around in their environment. And this process, again, is called transformation. It transforms them by them just taking up bacteria in their environment. Last process is called conjugation, and this is basically how bacteria have sex, right? Sounds very hot and exotic here, but it turns out that there are these little plasmids, they're little circles that are in some bacteria that basically allow them to code for this thing called a pili or a penis, right? And that's basically the bacterial equivalent of a penis, okay? And what this bacteria can do is it can inject, actually I think this guy is the male over here with the plasmid, inject DNA into another bacteria. And it allows for them to basically pass on some of their DNA to another bacterial cell. So conjugation is a third way bacteria can basically pass DNA to other bacteria. They can recombine their DNA, they can increase genetic variation. So these are three ways that bacteria can, again, increase genetic diversity, because being asexual, they need some other way to kind of increase their genetic variation and allow them to sort of evolve and change over time. So quick introduction to bacteria, viruses, and even prions, and that's it for this one.